Okay, so we reviewed DNA replication, so today we're going to talk about transcription and translation. So the other type of nucleic acid we have is RNA. Remember we talked about DNA, and there's two types of DNA. There's nuclear DNA and mitochondrial DNA. So when you talk about RNA, it's similar to DNA in that it has nucleotides in a strand with a sugar and phosphate backbone. But if I look at this sugar and phosphate backbone, what do we notice about it? How many sugar phosphate backbone strands are there in RNA? Just one strand. So it's just like one side of a DNA ladder. So only one sugar phosphate backbone. It's single-stranded. And most of the time when we look for RNA, we find it outside the nucleus. But with the exception of messenger RNA, which is formed inside the nucleus from DNA, and then it travels through the nuclear pore out to the cytoplasm. So, but most of the RNA we see is outside the nucleus. So the sugar is different. There's an extra oxygen in that sugar, so the sugar in RNA is ribose instead of deoxyribose that we saw in DNA. So that's why it's called ribonucleic acid, not deoxyribonucleic acid. So instead of thymine as one of the nitrogen bases in RNA, we find uracil. So when you're doing that complementary base pairing and you're making an RNA strand, you're just going to drop a U instead of a T. So A bonds with U instead of A bonding with T as it did in DNA. So we're going to talk about the role of the three types of RNA. We're going to talk about messenger RNA, transfer RNA, and ribosomal RNA, because the next two processes we're going to talk about, transcription and translation, are necessary to make proteins. And we talked about in our last lecture the importance of proteins for enzymes, because all enzymes are proteins. Structural parts, we have protein channels in our membranes. Our muscle is made of protein. Collagen in our skin and bone, and ligaments are made of protein. So what do we tell our patients when they're coming out of surgery or recovering from illness, because antibodies are made out of protein, so part of our immune system relies on protein. What do we tell our patients then, do you think, when they're trying to order off the menu and they want to get back to having some strength and recovery from surgery or illness? What should they order? Protein, yeah, protein. And what do people want to order? Carbs and sugar, right? Because, oh, I'm not very hungry. I think I'll just have some toast. Or I'll just have some fruit. Some people think, oh, I'm in the hospital, I'm going to eat healthy, I'm going to order fruit. And that's great. There's lots of antioxidants and vitamins, right, in fruit. Wonderful. But they need protein to heal. So we have to encourage our patients to order protein. And sometimes they don't want to because they think it's hard to digest. They're thinking like an omelet or a steak, right? It doesn't have to be that fancy. You can say, how about a little bit of fish? Or how about some cottage cheese, or a hard-boiled egg, or a small scrambled egg? Something simple. Yogurt is a good source of protein. We need protein to make protein, because when our digestive tract breaks that protein down into the little amino acids, that's what our cells are going to do, and reassemble those amino acids into different proteins, right? It's kind of like Legos. I can take the Legos, which would be the amino acids, and I can make a car, a house, anything I want, right? It depends on the cell and what their job is. So those amino acids from my digestive tract, when they enter a pancreatic cell, the pancreatic cells are going to assemble those amino acids to make insulin, glucagon, and all the digestive enzymes that the pancreas is in charge of, right? So it depends on the cell. So proteins, hugely important, and DNA has the recipe for that, and RNA helps take the recipe for making proteins from DNA in the nucleus out to the cytoplasm, to the organelle that's responsible for making proteins. And what organelle did we say is responsible for making proteins? Ribosomes. So the job of messenger RNA, which is the first molecule we're going to talk about, is to copy DNA in the nucleus and bring it out to the ribosome so we can make proteins. And that process is called transcription. So transcription is when we're making messenger RNA from DNA. So where does transcription have to occur then? If you look at this picture, it occurs in the nucleus. So we're going to unzip this DNA. So what enzyme do you think is required for that? Helicase. And then we're going to make RNA. So what enzyme do you think makes RNA? RNA polymerase, dropping nucleotides to make that polymer, that long strand of nucleotides, which we call messenger RNA. So then we can see here in the diagram that messenger RNA, follow the arrows, leaves through the nuclear pore, goes out to the cytoplasm and hooks up with the ribosome. So you can see the blue strand of messenger RNA that's attached to the ribosome, and that's where protein synthesis occurs. So the process of taking that messenger RNA and linking amino acids to make a protein is called, according to the diagram, translation. So when you think of a translator, right, someone who's converting one language to another, that's what we're doing here. We're taking the language of nucleic acids, of messenger RNA, and converting it to the language of proteins. So we're linking amino acids to make a protein. So looking at enzymes, same enzymes are required, except it's DNA polymerase, it's RNA polymerase coming in instead of DNA polymerase to make that messenger RNA. And again, notice that that messenger RNA is a single-stranded molecule instead of double-stranded. And we use the term polypeptide here. Polypeptide just means it's a long chain of amino acids that is a protein. It will become a protein. But there's a couple of things that have to happen first with the protein. Once it leaves the ribosome, in the cytoplasm, or on the rough ER, it enters the Golgi apparatus, and it packages them and finalizes their molecular structure. So proteins have a primary, secondary, tertiary, and quaternary structure, which is based on bonding that occurs within the polypeptide before it becomes a functional protein. So we're not going to go into the details of that in this class, but it talks about it in your textbook. So that's why it's called a polypeptide and not a protein as of yet. It will become a protein. So if we look at the diagram here, this is in your textbook. I'll give you a page number for that. I think we're in the 100s of the textbook for this. This is on page 102 of your textbook if you want a reference page for that. So the starting point for transcription, I should say, instead of a RNA primer, we have a little different name. It's called the DNA promoter. So the DNA promoter is where the starting point is for transcription. And when we look at translation, we're going to find that that spot corresponds to a special amino acid called methionine, which is the starting amino acid to start the beginning of transcription. That's the beginning of our protein. Because when you look at a long strand of DNA, there's little segments that code for proteins. And that little segment is called a gene, right? You've heard of genes, right? G-E-N-E. So that segment on the DNA molecule that codes for a protein we call a gene. And there's a bunch of nucleotides in between there that's just junk. It doesn't code for anything. So this transcription process of messenger RNA, we have to know where to start on the DNA molecule. So that's why that promoter is important. So we know where to begin, and we get that functional part of the DNA molecule, which we call a gene. Okay, so we start with initiation. So DNA polymerase binds to the promoter, separates the strands, and begins that messenger RNA synthesis. And it's the same process as we saw with DNA replication, as we're just dropping nucleotides. But the nucleotides are different, and then it's RNA nucleotides. So the only major difference is what nucleotide is there. Uracil. So you have a phosphate and a sugar with a uracil attached rather than a thymine attached. But everything else is the same. And where do these nucleotides come from that are floating around the cytoplasm of our cells? Yeah, from the digested products of our foods that we eat. Anytime you eat plant or animal matter, you're eating DNA, you're breaking it down, your digestive tract is breaking it down into nucleotides, and they're absorbed into our cells and available for RNA synthesis. So RNA polymerase continues on the strand until we reach the end of that gene, And then that piece of messenger RNA is removed, and you have a completed messenger RNA strand, again, that codes for a protein. And again, it's a gene, the part of the DNA that is the length of that protein, the recipe for that protein, again, we call a gene. Okay, so now it's going to leave the nucleus through the nuclear pore, and it's going to bind to a ribosome. So a ribosome, and if we look back up, let's see, there's a really nice diagram of this whole process on page 106 and 107. Kind of looks like this here. I'll just jump to this right now. So the messenger RNA down here, we can see leaves the nucleus through the nuclear pore, and it binds to a ribosome. So this ribosome is actually a coiled form of RNA. So this is the ribosomal RNA that we're talking about, the rRNA. That's what the big orange blob represents, is coiled up RNA. So the messenger RNA binds to the ribosome, and then we have these transfer RNAs that pick up amino acids based on the nucleotide sequence, the 3 nucleotide sequence on the transfer RNA molecule. So if I look at this diagram here, I can see this blue transfer RNA has it's all nucleotides. It's made up of all nucleotides, but there's a set of 3 nucleotides that codes for a specific amino acid. So this amino acid here, MET, which is methionine, is floating around in the cytoplasm. It binds to a transfer RNA based on the nucleotide sequence that's specific for that amino acid. And remember, that amino acid got there through digestion, right, and absorption into our cells. So then it picks up this amino acid. And this sequence, this 3 nucleotide sequence, these are the nitrogen bases of the transfer RNA, is called an anticodon. And it's going to bind to the messenger RNA through base pairing that matches that. So if the transfer RNA is UAC, if I base pair UAC on transfer RNA, what is that going to bind to on messenger RNA? AUG, which is the start codon for a gene. Methionine, that amino acid, is the start codon for a gene. So that's going to start the process. So we look at a chart. Now, how do you know that it's methionine? Well, there's a chart in your textbook. Sometimes it's in the chapter, other times they move it to the end. And I believe in this particular edition here, it's in the end of your chapter, or in the end of your book on page... If you find it before me, feel free to shout out the page number. Amino acid chart. Where are you? All right, page 103 in your textbook has this chart of amino acids. So we said AUG was on the messenger RNA strand that would correspond to the anticodon of a tRNA of UAC. So when we look at this chart, it's always using the messenger RNA strand for the codon. So the 3 base sequence on a messenger RNA that corresponds to the 3 base on the transfer RNA, it's called a codon if it's on messenger RNA. It's called an anticodon if it's on transfer RNA. So AUG here stands for methionine. So that's what this corresponds to. So this whole chart is, these are all amino acids. They just use the first 3 letters as a abbreviation. So this is phenylalanine, leucine, isoleucine, valine, alanine, threonine, lysine, arginine. Those are all amino acids. And we know that we need amino acids, right, for metabolism and, you know, the essential amino acids, because this is what we need to build the proteins for metabolism. Okay. So going back here then, So translation then, it occurs in the cytoplasm because we're at the ribosome. Transfer RNA, messenger RNA, and ribosomal RNA are required for translation. And the end product, what is the goal of translation? And transcription for that matter. What are we trying to get at through these processes? We're making a protein, yeah, polypeptide, which will become a functional protein. So when you look at transfer RNA, like I said, you know, it's coiled up RNA, but it has a special 3-base sequence here at the bottom. You can see CUC corresponds to GAG on the messenger RNA molecule, and it brings that amino acid. And in this case, the amino acid is glutamic acid, GLU, and it's bringing it to the ribosome. So this interaction here of transfer RNA to messenger RNA is occurring on the ribosome. So it brings these different amino acids in, and then we have at the end multiple amino acids from multiple transfer RNAs bringing those anticodons in, hooking up to the messenger RNA, and we have all these amino acids that are bonded to each other. What do we call the bond between two amino acids? The peptide bond. Yep. So that's what forms at the end of that process where transfer RNA brings in messenger RNA. So when we look at steps, we have initiation, where the transfer RNA brings in the amino acid to the ribosome. Then the codons are recognized where that bond is formed. Peptide bond is formed between those two amino acids. And then we shift down the ribosome to the next spot, adding more and more transfer RNA. So the different steps are initiation, elongation, and translocation. So that's kind of the order of major steps. So I think the best thing to do, as always, when we're talking about processes is to watch an animation. So let's go ahead and do that.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called Intron Splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion and the processes of transcription and translation make the production of proteins possible.
So I think that's an excellent animation. I think it really does a nice job kind of showing how all the different pieces fit together. So as far as, you know, vocabulary, there's a lot for our test. There's only 25 questions, so that's the good news. But we have to be power-packed in how I, you know, test on all that information. So like I said, I'll make a Quizlet to kind of get you familiar with the key concepts and reviewing. And what's nice about the Quizlet is you can make flashcards with it. You can choose different ways to learn. You can do like a multiple choice test. There's a lot of different options in Quizlet. So look for that in Blackboard. So let's go back to our PowerPoint and finish up the last couple of slides here. So translation then, like I said, the different parts, like the steps I should say, is initiation. And then she talked about the different sites in the video, the E, the P, and the A site. Do you remember what those different sites were responsible for? So what happens at the P site? Okay, that's where the binding occurs, right? Binding occurs at the P site. All right, so the transfer RNA binds to the messenger RNA unit, and then it slides down, right? So then we look at elongation. What happens in the E site? Do you remember what they said in the video? What happens in the E site? If we look here, what? Yeah, the transfer RNA is released from the messenger RNA in the E site. So it binds to the P site, is released in the E sites. That's kind of like, think of it as the exit site. That's where the transfer RNA is released. So it drops off its amino acid, right? And it slides down and leaves via the E site. So then the next codon to be translated is now in the A site. So that's where we're going to bind to the next transfer RNA. So this is where you're getting ready. That's the site of the messenger RNA that's getting ready to bind, but the actual binding is in the P site. And then this process continues until we get to the end of our, what do we call this section of DNA that's been transcribed via mRNA? What do we call this important strand, this piece of DNA that codes for a protein? Gene. Okay, so when we get to the end of that, the end of our gene always is going to have one of, this is a stop codon, one of these three codons on the messenger RNA. So again, it's on the messenger RNA. Whenever we see UGA, UAA, or UAG, if I look at that chart I showed you that was online, did I keep it? I don't know if I have it still. Let me see. No, I got rid of it. But when we get to these three codons, if I look at the chart on page 103 of your textbook, it corresponds to stop, which means end this growing protein here. Okay, so that's the end of it. So when that happens, this process stops, and peptide bonds are formed between the amino acids, and we have a complete protein, getting ready to be a protein, right? But then there's two other organelles, like the video said, that this has to pass through before it's a complete protein that's in the correct shape, because three-dimensional shape is really important for some of our proteins, like hemoglobin has a very specific shape in order for it to function properly and bind oxygen in the red blood cell. So what are those two organelles? Ribosomes make the protein. Well, some of the ribosomes are on the surface of this special organelle. The rough ER, so it modifies it, changes its molecular shape, then it's packaged, sent via exocytosis to the Golgi apparatus, which finishes the packaging, puts it into a vesicle, exocytosis out the plasma membrane, or out of the Golgi apparatus, then out the plasma membrane to wherever that enzyme is going to be used, right? So this process involves organelles. So this is happening on the ribosome. These two processes are happening on the ribosome, but it's not done until the rough ER and the Golgi apparatus has had their time to work with those proteins and put it in their final shape. So if we look at the process from the very beginning, this is a really nice slide. It kind of shows us how everything fits together. So on the DNA molecule, when we unzip it and expose the nucleotides, remember the pink represents the sugar phosphate backbone, and these are the nucleotides. So every 3 on a DNA molecule is called a triplet. And when I translate that to messenger RNA, those 3 are now called codons, right? When I formed my messenger RNA molecule. And then when that messenger RNA hooks up to a ribosome and transfer RNA bonds, the 3 complementary base pairs to the codon are called anticodons. And the anticodon corresponds to a amino acid. But when I look in the chart, what 3 nucleotides are we using? What does the chart correspond to? The chart in your textbook, for example, corresponds to the messenger RNA codon. So this is what we look up in the chart to find what amino acid to drop in that spot. And what's nice is your lab, we're going to do some of this in lab. Okay, we did a little bit of it last time. I handed out the worksheet, right? Did you see the DNA, the one where you had to transcribe, and we're going to do that next. Okay, so that is the anticodon that carries the start codon is methionine, right? That's the beginning. And we're going to continue on until we get to the stop codon that is one of those 3 stop codons, which is, like I said, listed on the previous page, UGA. Where did it? Yeah, UAA or UAG. Those are the stop codons. Now, if on the test I want you to do this transcription translation process, I don't expect you to memorize all the stop codons. That's factoid information that isn't going to affect your nursing care, so, or whatever program you're going into. So I'll give you the chart for that. All I want you to know is that methionine is the start codon, and that there are stop codons, and I'll provide you the letters for those. And everything in the middle is the recipe for making that protein. Okay, so let's look at the lab work.